So let's start up, guys, with the first image. And as you can see, this particular one, a given microbiological instrument is there used for the isolation of. So as you can see this particular picture, and if I zoom it up for you, it is seeing up written up gas pack. Okay, and gas pack, you know, guys, this is a chemical method of anaerobiosis, and this chemical method of anaerobiosis is used to create what? Anaerobic environment. An anaerobic environment is used to grow up which particular organisms? That is the anaerobes. Okay, and which of the following is anaerobe here? Clostridium. So everybody knows it. The Clostridium is the anaerobe, and it grows up with the help of this gas pack. And gas pack actually is like a ORS sachet. When you cut it open and keep it into a jar, it will absorb up all the oxygen and it will create what? Anaerobic environment. Okay. So try to tell yourself also the answers and all, and I will be telling you in and around that particular thing, a lot of things. So obviously it will be a very high yield stuff. In the Clostridium, guys, can you tell me up which is the culture media for them? Clostridium, anaerobes. What is the culture media? The culture media for anaerobes is what? Yeah, Dr. Anil, absorb oxygen, very right. Which is the culture media I want to ask for the Clostridium? Yes, anybody in the chat box? What is the culture media for this Clostridium? In which particular culture media you would love to grow this? Yes? Answer is RCM. And what is RCM? Robertson cooked meat media. What is RCM? RCM is Robertson cooked meat media. And this particular meat pieces, there was a question in the FMG exam, and this particular session is equally good for the NEET PG also. Okay, so if it is there, medicos, it is not a Loeffler serum. It is RCM, Robertson cooked meat media, and these particular meat pieces, okay, these meat pieces, if they turn pink, then that is termed as saccharolytic. Okay, and if the meat pieces turn black, that is termed as proteolytic. So that you must be knowing only. Okay, so Manisha, I've told up right, RCM. So meat pieces when turn pink, that is termed as, I am writing up here, saccharolytic. Okay, what is the meaning of saccharolytic? Which like use of the saccharides, that is termed as saccharolytic. Which use the proteins and all, that is termed as proteolytic. So saccharolytic is Clostridium perfringens. If you take up an example, Clostridium perfringens is a saccharolytic one, and proteolytic one is Clostridium tetani. Okay, so that is Clostridium tetani. Now this question already have come in the FMG exam, and in fact in one of the NEET PG also this have come, that which turns which color. If it turns pink, saccharolytic, and black, proteolytic. How to remember that? Pink sari, black pants. Okay, black pants, pink sari. So you will able to have some particular resemblance here regarding the meat pieces color change. An example I already told up. Fine. Yeah, very right, very right. Okay, so this is what is can be asked up in the exam again regarding the anaerobes. Okay, and answer to this particular question is Clostridium. Is it fine? Yeah, Clostridium tetani is slight proteolytic. You are right, Abhinav. But it does change it black. Okay. Now coming to this particular one. A 60-year-old man was brought in with a history of high-grade fever, okay, chills and rigors for the previous 2 days. He had a mild chest pain. Productive cough is also there. The sputum was submitted for investigation. The X-ray chest shows consolidation in the right lower lobe, a characteristic finding of a particular organism. The direct smear from the sputum showing this picture is being given up down. Okay, on the basis of gram staining, what is the probable cause of the infection? And I like to show you by zooming up this, that you can see up in this particular question, certain things are there which are in pairs. Okay, which are in pairs, and what is in the pairs, guys? Pairs is diplococci. Okay, what is there in the pair? That is the diplococci stuff, and means they are in pairs. And what is the shape of them? They look like flame shape, also termed as lanceolate. So what we are talking about, guys, what should be the answer here? The answer is Streptococcus pneumoniae. As everybody is telling, right? Answer is Streptococcus pneumoniae. And this picture, already a NEET PG question again can come. They can show you this particular one and this particular arrangement, diplococci arrangement, and this shape, also termed as lanceolate. Another thing which you need to know here in this particular question is this mnemonic. This mnemonic, I think, can definitely be asked, and that is bio. Okay, and what is there in the bio? Bio is, it is bile soluble. Okay, we are talking about what? We are talking about pneumococcus. Pneumococcus is P for P. It is positive, bio positive, bile soluble, inulin fermenter. Okay, this particular polysaccharide is being fermented by this, and optochin sensitive. And optochin is a drug, so it is optochin sensitive. Okay, so bile soluble, inulin fermenter, and optochin sensitive. This is what is termed as bio, and bio is positive in pneumococcus, negative in viridans. It was asked in NEET PG. Okay, so bio, these particular biochemical reactions, bio, as the mnemonic tells, this particular thing is negative in, it is negative in the viridans, and it is positive in what? It is positive in the pneumococcus. Okay, remember that. Now, another important thing, the most common cause of CAP, what is CAP? Community acquired pneumonia, is pneumo, guys. Okay, is pneumococcus. The most common cause of typical pneumonia, typical means lobar pneumonia, again answer is what? Pneumococcus. Okay, the most common cause of pneumonia in HIV positive, in HIV positive, again your answer is what? Pneumococcus. Okay, so all of them, answer is what? Pneumococcus. And the question which have recently came, that the most common cause of atypical pneumonia, the answer is Mycoplasma pneumoniae. Okay, and that pneumonia is termed as what? Walking pneumonia. And that is the atypical pneumonia, not the typical one. So in HIV positive also, overall most common is pneumococcus only, but when the CD4 count turns out to be less than 200, then comes pneumocystis cerevisiae. Okay, pneumocystis cerevisiae pneumonia at a CD4 count of less than 200, for which you use up which particular drug, guys? Cotrimoxazole. Okay, so very right, everybody is telling wonderfully. Okay, so these are very high yield stuff regarding the pneumococcus which ought to come in your exam. Another one, if I ask you that there is something about the capsule of this organism, what is that particular something? That something is Q-lung reaction. That something is what? Q-lung reaction, and this is basically the capsular swelling. Q-lung, lung, pneumo. So Q-lung reaction is associated with this particular pneumococcus only. So these are the important ones which you need to know regarding the pneumo. Now, again, another question which they can give you, and they can give the features of pneumonia in the question. Okay, and they can ask up the typical pneumonia features only, and they can show you this particular culture plate in the exam. Okay, can you tell me, guys, by seeing up this culture plate only, can you tell that what we are talking about? Yes, anybody in the chat box? What we are talking about, anybody can tell that this particular culture plate is showing up something, mucoidness. I am giving you a hint. There is a mucoidness in this, and mucoidness is a feature of which particular bacteria, guys? Mucoidness is a feature of which particular bacteria? Abdul is on the right track. Okay, Abdul in the chat box is on the right track. It is towards the Klebsiella, guys. So they can very well relate it with this particular picture, and they can show you this McConkey agar. This is McConkey agar, and on this McConkey agar, you can find mucoid colonies. Okay, and in the chat box, somebody is telling a lactose fermenter. Yes, it's a lactose fermenter, and Klebsiella is going on it, and this mucoidness is typical of Klebsiella. Okay, so as they have given the gram stain picture of pneumococcus, here what they can do up, they can give this, and they can ask up that alcoholic is there, and it is having the sort of consolidation and all. Okay, and a lobe involvement is present. Which particular bacteria? To Klebsiella pneumoniae. And along with that, the favorite of examiner are 2 points. Everybody knows them. I suppose you must be knowing regarding Klebsiella pneumoniae. Sommelier is not non-lactose fermenter. This is a lactose fermenter. This is Klebsiella pneumoniae is a lactose fermenter, not the non-lactose fermenter. Okay. And two particular points which I am telling up is what? They are red currant jelly sputum and bulging fissure sign, which is seen on the chest X-ray. They are so favored examiner. The examiner must have had some time Klebsiella pneumoniae. It have become favorite of them. 20 or 25 times repeated question this is. Red currant jelly sputum and bulging fissure sign is a feature of. It is a feature of Klebsiella pneumoniae. Is it fine? So they can easily relate up with this and they can ask you this particular question. Now you can say, is the E. coli can also show these types of pictures? No, mycoidness will not be there in E. coli colonies. Lactose fermenter it is, but mycoidness will not be there. Mycoidness is a feature of Klebsiella pneumoniae. Okay, and if we are talking about Klebsiella, guys, remember these particular things. Regarding the Klebsiella, important thing is Klebsiella, illa, okay, it is non-motile. Illa it is there, so it is having non-motility. It is the important feature of the Shigella, Klebsiella. They are what? They are illa. They are non-motile. Fine. So remember that particular thing also. Yeah, Arvind, very right. Red currant jelly sputum, bulging fissure sign. Otteri, Dr. Anil, you have told everything regarding it. Wow. So gram-negative, non-motile, encapsulated, lactose-fermenting, facultative anaerobe, rod-shaped bacteria. Okay, you can add one thing more in that if you have written so much. You can add upon it is urease positive. Okay, it is urease positive and it is the one which produces mucoid colonies. One of the common nosocomial infection, this particular fellow is there in that. Fine? No, it's not swarming growth, Chandni. You are wrong in that. Fine. So this answer is Klebsiella pneumoniae, and other one which are important, let me tell you, Klebsiella uzini leads to uzina, that is atrophic rhinitis, rheumy nose and immense foul smell. Anosmia is present in that particular Klebsiella uzini infections, and one is Klebsiella rhinoscleromatis, which leads to rhinoscleroma, that is termed as woody nose. And when you go for the biopsy of it, you find what? You find Mikulicz cells and Russell bodies. Favorite question again of the examiner. Okay. Normally harmless. Normally harmless. Yeah, you can say they are commensals in some bodies, yes, but you can't say normally harmless it is. Now, coming to this particular one, guys. Identify the hemolysis and the bacteria producing it. As you can see this particular picture, if this comes in your exam, can you tell me what we are looking up to? Merciful. Yeah, very right, Manisha, very right. Now coming to this one, yes, tell me what you are observing in this. Aren't you observing a double zone of hemolysis in this? So this is termed as double zone or target hemolysis. But the important thing is target hemolysis, double zone hemolysis of what? What is the options telling up? Yes. No, Janni, it doesn't show swarming growth. No, it is having a mucoid one. Okay. So tell me this particular one. Double zone of what, Preeti? Double zone of what? Is it double zone hemolysis by Clostridium tetani? Is it target hemolysis of perfringens? Is it beta hemolysis of Botulinum and incomplete hemolysis by Clostridium difficile? Yes, tell me fast. Double zone is fine. Answer is what? A, B, C, D. Yes, what is the answer? Preeti have told A and answer is wrong. Okay. Clostridium tetani does not show double zone hemolysis. Which particular organism does show it? The answer is B, guys. Answer is B. Target hemolysis it is, and shown by which fellow? Clostridium perfringens, which is also termed as Welchii. Okay. So answer to this question is B rather than A. Like this, the examiner will play the tricks, because double zone is also termed as what? Target hemolysis. And another important names of the reactions of this is, already we know in our lectures, senior NTR. This is the mnemonic for it. The reactions of Clostridium perfringens, Welchii. S is stormy fermentation of litmus milk. N is Nagler's reaction. T is target hemolysis. R is reverse camp test. Reverse camp test. Okay. So answer to this question is B. Answer to this question is B, and let me revise up very fast the Clostridium. Clostridium perfringens, what does it do, guys? It does gas gangrene. Okay, gas gangrene, Clostridium perfringens, and it is a what? Which type of bacteria? What is the shape of it? Brick shape. Okay, brick shape. If the boat shape comes, then that is septicum. Okay, Other than that, it causes food poisoning. An important point regarding food poisoning is it is the only one which produces heat labile antirotoxin. Otherwise, all others, they mostly produce heat stable. Okay, that is important point. And there is one particular thing termed as pig bell. And pig bell, it is, as the name suggests, eating up uncooked pork. That leads to this. And beta toxin type C does that. Okay. Clostridium tetani. Clostridium tetani, the important question which can come regarding that is this particular one, the tetanospasmin and tetanolysin, which is important, spasmin, and spasmin acts where? Pre-synaptic inhibition of glycine and GABA. Okay, that leads to the constant stimulation. And can you tell me which particular snare protein is involved in this? The snare protein involved is synaptobravin. Okay, synaptobravin. Clostridium botulinum. Clostridium botulinum is the one which have an action of anticholinergic, decrease in acetylcholine at various junctions like neuromuscular junctions, like postganglionic parasympathetic. Okay, so number of other junctions also it has having action, and the main action is what? Decreasing up the acetylcholine levels. Okay, it leads to canned food poisoning, that you know, antirotoxin is responsible for that. Most of them are neurotoxins, and this particular one, important regarding this is that it is being used for the Botox treatment and all, and most potent toxin on earth that is being told up is Clostridium botulinum. And snare proteins, synaptobravin, syntaxin, and SNAP25. These names, they are coming in your exam. SNAP25, syntaxin, and synaptobravin, these are the snare proteins involved up for the Clostridium botulinum. Okay, telling up regarding flaccid paralysis, Abdul, very good. Okay, telling up Clostridium difficile, also termed as Clostridioides difficile. Fine. CDAD, associated diarrhea, mostly run by which fellows? Third generation cephalosporins, then Clinda, then ampicillin. Which is the drug of choice now for this? That is pseudomembranous colitis and all, fidaxomicin rather than the vancomycin. Fine. And what is the best way to diagnose this particular stuff? The best way to diagnose this is toxin demonstration by ELISA, along with that GDH. And what is GDH? Glutamate dehydrogenase estimation. So we have discussed up the full Clostridium in 5 minutes. Fine. So I suppose this particular thing will help. These all are questions and they will be coming up in exam. Fine. Now coming to this. See this particular picture and tell me, obviously it's written up there itself that which test I am talking about, ALEC test. So my question for you is that this particular test is done for which bacteria? First of all, and out of these particular ones, can you tell me which should be the answer? That obvious hai. This is not the answer. This is not the answer. This is not the swarming of it. So obviously answer is this. This is a agar diffusion in 2 dimension. So guys, this is Ehrlich gel precipitation test, which is not done nowadays, but definitely can be asked. Okay, and this particular thing is which sort of a diffusion? Double diffusion in 2 dimensions. What it is termed as? Double diffusion in 2 dimensions. And double diffusion in 2 dimensions, typical example if they ask is Ehrlich gel precipitation test. Ehrlich gel precipitation test, which is done for what? Corynebacterium diphtheria. Okay, for the diphtheria diagnosis, you do this particular one. And if somebody asks you which type of diffusion? Double diffusion in 2 dimensions. Means in 2 dimensions only you can measure. 3D you can't measure. So that's why you say it. And what is it? Toxin antitoxin action. It is toxin antitoxin action. It's precipitation lines they produced up. As you can see up in this, these lines they are there. So non-toxigenic strain you take in that, positive control. Then the patient isolate you take, and a known non-toxigenic Corynebacterium diphtheria you take. Okay, and what you observe is when this particular filter paper strip with the Corynebacterium diphtheria antitoxin you take and you produce up these 3 lines, positive control, negative control, and the patient line, you observe the formation of these precipitin line. Okay, so as the precipitin line are produced in patient isolates also like this, that suggests yes, patient is having an attack of toxigenic stuff. It is producing up toxins. Is it fine, guys? So this you need to know and see this. It is non-toxigenic, negative control, and it is not producing any sort of precipitin line. So this is used for the toxin demonstration. Is it fine? And regarding diphtheria, I like to tell you 1 or 2 important points, guys. And those important points are this way, that every CD is not pathogenic. It is made pathogenic by beta-phage tox gene. And this conversion is termed as what? Lysogenic conversion. And lysogenic conversion is an example of transduction. And whenever you heard of this transduction, what should come in your mind? this particular bacteriophage or phage, whatever you call it. Okay, and this was that FMG question only, last to last time. Bacteriophage mediated transfer is termed as what? The answer is transduction. Okay, so this is what is the answer. And if we talk about the culture medias and all, early diagnosis, Loeffler serum slope, like definitive diagnosis, potassium tellurate agar. Okay, and this dictum you already know that it is having pseudomembrane, and when you try to plug it up, that bleeds. And this particular is having this bull neck and stuff and all that. Okay, so that you already know. Besides this, this dictum is also important. Diphtheria is dash, not dash. It's toxemia, not bacteremia. So that's why we are demonstrating the toxin here in this particular one. Okay, guys. So these all are important stuff regarding diphtheria. And along with that, I like to tell you regarding non-corny diphtheria also. And important one is Corynebacterium minutissimum, and that leads to erythrasma. Okay. And that is having correlated fluorescence. You must have read in the skin also that particular thing. And we have made to remember it in this particular stuff, like Corynebacterium minutissimum, Minu's mom prepared up red rasma, that is, or red rasam. So, and any sort of that erythrasma. So, important question. Very good, Dr. Anil. Albert, Abdul, yeah, right, very right. Albert stain is there. Chinese letter pattern is famous for it. Green colored one if it is Albert. Otherwise, you are having this purple stuff. It is a gram-positive bacteria. It's a gram-positive bacilli. So it can take up both the stains. Okay. So this is regarding the diphtheria. Antidiphtheric serum. That is the treatment of choice. Now, guys, coming to this one. Can you tell me what you are observing in this particular picture? Up. Tell me this. What you are observing in this? Is it a gram staining? Is it Zn staining? And if it is Zn staining of what we are observing this? Tell me this answer. Yeah, Hare Krishna, very right. Chinese letter, cuneiform arrangement. Yeah, tell. This particular one, please. Zn staining or gram staining? What it is, guys? Tell me. Zn staining or gram staining, first of all? Yes, anybody? Yes, please. The comment section. Yeah, SM, it's very useful for NEET PG also. Yeah, 24 SM. Don't miss it. C. The answer told by the students is C, and they are right. It's Zn staining, and it is for the Mycobacterium TB. And you know that Zn staining, what is the steps of Zn staining? Carbonyl fission. And the mnemonic for that is car inside se hai blue, means carbonyl fission. Then you do the intermediate heating, and then 20% H2SO4 in this particular case, and then you're using of what? Methylene blue. Okay, so background actually not the proper blue, it is not a proper stain, but these particular things, they are pinkish to reddish in color, and they are arranged like that. We are typically talking about what? Zn staining. And for the gram staining, the mnemonic is what, guys? Methyl gas. So that also you need to know. Methyl violet, okay, grams iodine, acetone, and safranin. Okay, gram positive bacteria, that is, they look like purple to violet, and gram negative, pink. Why? The most important factor which governs the gram staining is peptidoglycan thickness. And peptidoglycan thickness, there in the cell wall is whom? Gram positive or more thick. More thick cell wall is the feature of gram positive bacteria. Okay. The question which have come recently is, in your exam only, in the FMG, this was there, that lipopolysaccharide, that is a feature of which cell wall, first of all? The answer is gram negative. Okay. And from that, there is an antigen which comes out, that is termed as O antigen. Now, what is O antigen? Somatic antigen. Okay. O antigen is what, guys? O antigen is somatic antigen. Okay. Dr. Anil, you have done the research, huh? Are you a microbiologist? Slow growing, chemo, organotropic, non-motile, non-spore forming aerobic bacillus. Oh, my. Good. Yeah, very right, Abdul. Very right, Vikas. Negative, what negative? What negative? Preeti, what negative? Answer is C. Okay. What is full form of Zn, guys? Ziel-Nielsen. Okay. And who is the actual discoverer of this particular acid fast staining? That is Paul Ehrlich. Yeah, very right. O antigen is somatic one. Now, guys, tell me this, the famous culture media, which you should know. Without knowing this, you should not go in the exam. Trick से याद किया हुआ है, ओ हो, लेकिन यार उसकी ज़रूरत नहीं है अनिल, इतना याद करने की ज़रूरत ही नहीं है भाई, नो नीड. एक्चुअली प्रॉब्लम पता है कहाँ है? प्रॉब्लम ये है कि वी शुड ऑल्सो नो दिस कि what should not be learned. You are not sitting in the MD exam यार, तो why you are need to remember everything, chemo, organotropic, slow growing and all that. So no need, I suppose. But as such, if you are able to remember it, good, no problem. Yeah, tell me, which particular media, guys? Without this, you should not go in your exam. NEET PG, INICT or any, which particular one? Vikas have told wonderfully, yes, the answer is LJ. LJ stands for what? What is the LJ full form? Lowenstein-Jensen media. Lowenstein-Jensen media. This is having how much percent agar? Every particular media is having agar, na guys? 2%. It is having how much? LJ media is not having agar. Okay, it is having eggs, which plays the role of solidifying agent. Okay, so that you first of all know. And because of that only, we inspissate it. Inspissation is being done for these LJ media, not the autoclave. Otherwise, for most of the culture media, what you do? Autoclaving. For this, you do what? Inspissation. Both are moist heat sterilization. Rough, tough and buff. Preeti, you're right here. Buff, rough, tough and buff. And how much time the colonies of this particular Mycobacterium TB comes on the Lowenstein-Jensen media? Can you tell me? No agar. Daffodil have come with no agar. Yes, you are right, daffodil. Daffodil used to be my house in my school. The flower. Okay. Yeah, rough, tough and buff colonies, they are present up 6 to 8 weeks it takes to them to come on this particular one, and it is a sort of eugonic or dysgonic growth. The answer is eugonic growth, luxuriant growth, but it takes time to come. Now, here only I like to tell you regarding these C and D options. Okay, so Dorset egg media is also used for TB only. Nobody knows it, but it is true. Dorset egg is also used for what? Mycobacterium TB, and Pitra greeni media is also for the TB. Okay, Mycobacterium TB. But this you already know, Haemophilus serum slope is for what, guys? It is for Corynebacterium diphtheria. Okay, it is for Corynebacterium diphtheria. Okay जी, तो मैंने बता दिया, like culture media mostly autoclaved, but this particular one is what? This particular one is inspissated. And inspissation and autoclaving both are what? Moist heat sterilization. They are what? They are moist heat sterilization. Now guys, this particular table itself is full of questions. Okay, this is full of questions, and short, this particular question is coming regarding the E. coli pathogenesis. Okay, so let me tell you regarding the important points here. Okay, I will just select up the important points. Yeah, Kartavya, you're right. See this particular one, guys. First of all, see this Fimbria. This was a NEET PG question also. Fimbria helps in what? The Fimbria helps in adhesion. First point. Okay. And this particular P. Fimbria, this is there in certain E. coli's which attaches to the uroepithelial cells. And they leads to what? They leads to UTI. Okay. That leads to what? That leads to UTI. And this assists in what? Urinary colonization. That you already know. So P. Fimbria, they helps in adhesion. Now talking about pathogenic, enteropathogenic E. coli. So guys, P for P, that is pediatric age group diarrhea. It causes diarrhea in the nurseries. That is one of the points. Another important point regarding this particular fellow is that it leads to A/E lesions. And what is A/E lesion, guys? Attaching. It attaches to the intestinal alveoli and damages them, effaces them. So that is termed as A/E lesions. That is done by enteropathogenic E. coli. Okay. Then you see this side, guys. CFA, colonization factor antigen. You're observing this colonization factor antigen, and this particular CFA is present in ETEC, enterotoxigenic E. coli. This question can come this time, and when I say this question can come, usually it comes. Okay. So CFA is colonization factor antigen, and this is present in what? This is present in ETEC, and ETEC is toxigenic E. coli, which is one of the common causes of what, guys? Traveler's diarrhea. Which is the common causes of what, guys? Traveler's diarrhea, it is the most common cause of that. Traveler's diarrhea. Which one? ETEC. And you will be having an interesting aspect here that this is the most common cause of diarrhea in children if bacteria is asked. Pediatric is fine, but overall in the children, if somebody asks you, as per the bacteria, ETEC. But overall, obviously, rotavirus is there. But bacteria, if it is asked, then it is ETEC. Abdul have told, right, travelers. Now, guys, these are the toxins, ST toxins and LT toxins of it. That is stable toxin and labile toxin of the ETEC, which causes diarrhea. And stable toxin, I already told you in my lectures. Stable toxin acts through which mechanism, guys? Increase in cyclic GMP, guanosine monophosphate. Okay, stable में कौन रहता है? In the stable, what resides? Horse. And what you call horse in Hindi? Ghoda. Okay, increased cyclic GMP is for the stable toxin. And this particular same thing goes where also? Bacillus cereus. Bacillus cereus hematic strain, Bacillus cereus diarrheal strain. Yes or no? Manisha, very right. Rota, you have told. And travelers, you have told. Okay, so increased cyclic GMP is stable toxin. Labile toxin is increased cyclic AMP. That is adenosine monophosphate. So these two things you need to know. Now, virotoxins. Another question which is there likely coming in the exam is this. Virotoxin associates with HUS, guys, and the bloody diarrhea. And which particular one is there? EHEC. Okay, as it written also. Virocytotoxin or virotoxin. Also termed as Shiga-like toxin. Okay, it is also termed as what, guys? Shiga-like toxin. Fine. So this is also there. Besides that, what is the culture media? The culture media for this particular is SMAC. And what is SMAC? Sorbitol McConkey agar. Okay, sorbitol McConkey agar is the culture media. This was asked in NEET PG this time. And the next question might be that which other media is used? The answer is rainbow agar. So SMAC and the rainbow agar, that is there in this particular one, which you need to know. Okay, and as you can see that certain E. coli, they having capsule also, certain ones, not everybody, certain ones they having. And as we have talked about the lipopolysaccharides, lipopolysaccharides, they lead to the release of endotoxins. And endotoxins leads to what? Endotoxic shock. So that is also there. And those who have this capsule and this K1 antigen, the capsular antigen, they enters into brain and they leads to this neonatal meningitis also. And this is explained up that these are interleukins which are released up and the patient lands in what? Endotoxic shock. So this particular flow chart itself is telling up a lot of questions regarding the E. coli, and we are talking about the diarrhegenic E. coli mostly, and here we have talked about the UTI also. Okay, here we have talked about the UTI also, and one of the questions NEET PG was an INCT. What are the screening tests for the UTI? Screening test for UTI is grease nitrite test, okay, then leukocyte esterase test, and catalase test. These are the screening tests, and obviously definitive and confirmatory is culture. And culture is done on which media, guys? CLED. And what is CLED? Cysteine lactose electrolyte deficient. Okay, why not McConkey agar? Because McConkey agar is selective, CLED is non-selective. Okay. Very good, very good, very good, very good. Namaskar, namaskar, namaskar. Chale aage? So I suppose you must be knowing up a lot of things now. Now, this particular one, guys. Choose the right statement on the basis of given media, and as you can see this picture, if I zoom it up, so these are jet colored colonies, and jet colored colonies, they are of Salmonella typhi on which media, guys? On which media we have this jet black colonies? So jet black colored colonies of Salmonella typhi on which media? Wilson and Blair. Wilson and Blair. Wilson and Blair is having jet black colonies of Salmonella typhi, and this is what is the picture of this particular one, which you should know. Okay. Otherwise, if we talk about the various culture medias here, the transport media is SACS. This was asked. Okay. Enrichment media is cell-line broth, and the best selecting media is Wilson and Blair. And the same medias exactly they are used in the Shigella, but Wilson and Blair is exclusive of what? Salmonella typhi. Okay. And the Salmonella typhi have come here. So let's talk about Salmonella typhi also. The question of last FMG was that in which particular week, which particular test is done. So first week, second week, third week and fourth week. And remember, this is week. And they have asked you on the third day, which is the best test to be done. So obviously on the third day, it is the blood culture, which is best. And there is bone marrow culture, which can be done if you have that particular capacity also. Otherwise blood culture. And the question asked was third day. And third day obviously comes in the first week. The answer of R should be what? Blood culture, but everybody have written stool because they were not knowing that this is weak. Okay, and the question asked regarding what? The question asked regarding the day. Okay, so A is agglutination, and agglutination is which test? That is the Vidal test, which nobody does it. Now they go for typhoid hot and all. Okay, S is stool, that is stool culture. U is urine culture, which is actually not done proper. ELISA, that is being done, urine ELISA, along with the urine culture also in the fourth week. Okay. Yeah, very right, very right. Very good. So this is what is the mnemonic for this Basu. And another question which can come here is this particular Faggot sign. And Faggot sign is relative bradycardia, you know. Okay. Step ladder pattern, rose spots, okay, pea soup diarrhea. They all are features of what? Salmonella typhi typhoid fever. And what is the incubation period, guys? This particular incubation period is definitely 1 or 2 questions in NEET PG as well as in the FMG exam. And this on my YouTube channel by my name only, I have made a video of this. So you can see that incubation period one. Definitely 1 or 2 questions might be coming from that. I will share the link. Okay. So this is there, guys, and important is that in this, the leukocyte count does not increase as other fevers. It remains normal or it goes down. So this is also a characteristic point of typhoid fever. Okay. And the culture media is already talked about, and drug of choice you already know, that is the ceftriaxone. Okay. Yeah, Preeti, very right. Now tell me, guys, about this particular picture. Greenish color picture, and antibiotics are seeing in this. What we are talking about and which media it is. Yes, please tell. Manisha, very right. This question is coming. Manisha, I was telling now. Osteomyelitis in sickle cell anemic patients. Yes, Salmonella. Salmonella, certain species, they can definitely does the osteomyelitis. They are the most common cause of osteomyelitis in sickle cell anemic patient. And this question is coming. Manisha, I have told it wonderfully. Yes. Okay. So this particular antibiotic testing is being done for the Pseudomonas, which have turned this MHA plate also green because of the pigment pyoverdin. Okay, and now if we are talking about Pseudomonas, in the Pseudomonas, the first question which comes up is that what is the culture media? The culture media is citrimide agar. Which particular important conditions associates? Important is malignant otitis externa. Common in whom? In the DM fellows. Important here, ceftriaxone and vancomycin is not anti-pseudomonal, first point. Okay, and polymyxin B and colistin. In fact, polymyxin B only is remaining now, against which no resistance is reported up till. Okay, so that was also being asked. Burkholderia malae, Pseudomalae, Burkholder capacea, they are the brothers of Pseudomonas. They come in the family of non-fermenters. Okay, Pseudomonas is what? It's catalase positive as well as oxidase positive. But one guy which is nowadays having a swag, that is Acinetobacter baumannii. That is oxidase negative. And it is found where? In the ICU. And it is multi-drug resistant isolate. Okay, so these are the important ones. Yeah, very right, Abdul. Burn wound. The favorite of that particular one is burn wound. And Arvind is telling up this is KBDDT. Very right, guys. This is Kirby-Boyer disc diffusion testing on MHA. And if MHA is not there in the option, the second best answer is Na. That is the nutrient agar. Is it fine, guys? Swarming is a feature of Proteus. Naglers is there in perfringens. Dynes phenomena is there in Proteus. Okay, P for P. Now, this just now we have talked about oxidase 1. And oxidase is actually what I should tell only. Okay, oxidase is this white color changes to blue. This white color, the strip changes to what? Blue within 10 seconds. Okay, this particular one changes in blue within 10 seconds. and this is what is termed as oxidase positive. And the bacteria which have just gone, it is strongly oxidase positive and catalase positive both. Okay, catalase and oxidase positive both. So answer to this question, can you tell me? Except. So guys, this is slightly tricky question because you need to know a lot of things regarding this. But I should tell you one thing, if you are smart enough to exclude other things, then you can easily exclude up that E. coli. E. coli is an Enterobacteriaceae member. Okay, and Enterobacteriaceae family is fully oxidase negative. Okay, so answer of ours is obviously E. coli, and smartly you have reached to that because Brucella is oxidase positive, Vibrio is oxidase positive, and Neisseria both, gonococci, meningococci, they both are oxidase positive. Okay, they both are oxidase positive. Yeah, very right, very right. So answer is D, E. coli. Okay, and this is the oxidase test. Now guys, if you see this particular picture, we zoom in, you can see that in between there is no staining and the poles are stained. So which particular staining we are talking about, in which the poles are stained and in between it remains unstained? What we are talking about, guys? Kartavya, it's better to remember this particular thing, that which are oxidase positive. Okay, because this word is oxidase negative. Yeah, Preeti, you are very right. Answer is Yersinia only. But what is the answer? A, B, C, D. Which is the answer here? A, B, C, D. Bipolar staining. Answer is, yes, answer is, answer is C. Giemsa stain. So you can stain the Yersinia pestis by the Giemsa also, and this have come in this NEET PG exam. Giemsa stained. And if it is for Giemsa stain, one more stain name you should know, Wason stain. Wason staining and Giemsa staining, the characteristic done for Yersinia pestis and this particular Yersinia pestis. Can you tell me the culture media for this? What is the culture media for this? What is the culture media for Yersinia pestis? Yeah, in the comment section. What is the culture media for Yersinia pestis? Ghee broth. Okay, what is the culture media? Ghee broth, and stalactite type of growth is there, and there are three types: bubonic, septicemic and pneumonic. Okay, and in this, mostly the question now they come in the form of this, that somebody have gone to the foot go down and there is darkness, somebody have bitten up and there is a swelling in the inguinal region, inguinal buboes, and when you go for this inguinal buboes smear, biopsy smear, you observe this safety pin appearance. Okay, so we are talking about this particular stuff, that is the Yersinia pestis. Yersinia enterocolitica. This is the new one which can be asked in the NEET PG and ICT also along with FMG exam. It shows appendicitis-like features. Okay, it shows appendicitis-like features, and important aspect is differential motility is observed by the Yersinia enterocolitica. Okay. Now coming to this particular one, another important question which can definitely be asked in the exam, and as it is written up, Haemophilus influenzae colonies are there and Staph aureus is being streaked off. If these two names comes, immediately what should come in your mind is satellitism. And this was again asked in the NEET PG. Satellitism is shown by which particular bacteria? The answer is Haemophilus influenzae shows it in the presence of Staph aureus. And satellitism is actually the presence of certain factors X and V near the streak of Staph aureus. That is what is termed as satellitism. Is it fine, guys? So satellitism is this particular entity, and this was asked by this name only, that we are talking about which particular organism? Haemophilus influenzae. Haemophilus influenzae is a gram-negative cocobacillus. First thing, loves to grow on the chocolate agar. The capsulated bacteria, they are more invasive, leads to what? Meningitis, epiglottitis, normal ones, LRTIs, okay, otitis media. They are being done by what? Non-capsulated one. So these you need to remember in this particular sense. Besides that, satellitism is shown by this. It is catalase and oxidase positive. Okay, so these all, they can be asked in some form or another regarding it. Okay, so answer is B, as everybody have told up. Now guys, this one, this everybody should see and tell that what culture media we are talking about and which colonies are there on this particular one. Then we'll talk about certain important points regarding it. Yes, can you tell me guys, which particular media? Yeah, Anil, tell me Vikas, tell me Daffodil, tell me Krishna, Cortex, Preeti, tell me. Which culture media it is? By seeing up only, if you are telling up, what is the answer? Is it XLD? Is it TCBS? Is it DCA? Is it CLED? What it is? And the Medico Journey is selling D. Arvind Kumar is selling B. Arvind Kumar again is telling TCBS. So guys, tricky one. Again, this is a tricky one. As such, what I want to ask up is this only, TCBS, thiasulfate citrate bile salt sucrose, which is a culture media, best selective media for Vibrio cholerae, and it is a greenish color media on which this particular yellow stuff, yellow colonies are coming up of the Vibrio cholerae. But you are also right guys, this can be CLED also. But what is the clause for that? It is not showing the growth of Proteus because Proteus is NLF. It is a non-lactose fermenter. It will not change the color to yellow. But yes, if instead of Proteus, what is being written of E. coli, okay, Klebsiella, which are lactose fermenters, Shigella soni, they will change the color like this. Then the question will become tricky one. Okay, but as such, the answer is TCBS here. And CLED, cysteine lactose electrolyte deficient, this is not the answer because we have told up regarding the Proteus, which is a non-lactose fermenter. But remember this also, CLED is also green color and it changes to yellow when the lactose fermenters grows on it. Remember that. Which one is better, CLED or McConkey? CLED is better because it is non-selective. Another NEET PG question. Okay, DCA, deoxycholate agar is used for Salmonella and Shigella. It is not of this color. Okay, it is not of this color. Now, as you can see this particular picture, the lesion is painful, bleeds easily while taking the swab for the gram staining. What is the probable cause after seeing of this particular picture? Is it Haemophilus ducreyi? Is it Trypanosoma pallidum? It is HPV or HSV2? Yeah, Dr. Anil, you are right. Rice watery stool, which mimics of which particular poisoning? Arsenic poisoning. And the question which have come recently is that the cholera toxin acts on tight junctions or zonooccludins. They are the same thing. Okay. And in that the B is there, the B subunit. It attaches to which particular receptors? GM1 ganglioside receptors, and it does ADP ribosylation of G stimulatory protein. That you need to know. And there is increase in what? Cyclic AMP. And you are right guys, the answer is Haemophilus ducreyi. Haemophilus ducreyi, dukh raha hai, it's painful and it bleeds. That is what is chancroid. And the point which I want to tell up here, when you take the biopsy from here and stain it with Giemsa, you will find what? School of fish. School of fish appearance or tram track appearance. That is observed in this particular one. Okay. And Trypanosoma pallidum, the same thing, is painless, and this particular painless stuff, along with that, it is a hard chancre over the genitals. Okay. In this, it is a soft sore, soft ulcer, which is painful. Krishna, you are telling dukh raha hai to chancroid. Yes, you are right. Now, this I have already told up. This is a repetition one. I have already told up this one. Ghee broth. Which particular organism loves to grow in ghee broth? So, do you know? Answer is Yersinia pestis. Okay. And the important point regarding Acinetobacter, which I have told you, it is oxidase positive. Sorry, this oxidase negative. And this particular Acinetobacter, why it is famous? Because it is found to be a MDR isolate in the ICU. Okay. MDR isolated where? In the ICU. बिल्कुल इम्पॉर्टेंट है डॉक्टर अनिल, इंस्टाग्राम के क्वेश्चन पोस्ट बिल्कुल इम्पॉर्टेंट है, वो देखो सारे के सारे. very right, very right. Answer is Yersinia pestis, and Acinetobacter you need to know is oxidase negative and MDR isolate hai in the ICU. And recent INI-CD question, this name at least you should know, Trophomonas multifila. By chance, if you get up this particular name in your paper, which you might get, Stenotrophomonas multifila, this is also a non-fermenter. Okay, this is also a non-fermenter, intrinsically resistant to number of drugs and susceptible to cotrimoxazole. This was asked in INI-CD, and this can be repeated again. Okay? Okay guys, let's move. Now, another favorite question. Everywhere darkness is there, and against that you are observing these particular fellows moving. So we are talking about what, guys? Dark field microscopy for the Treponema pallidum. And Treponema pallidum is a spirochete. Okay, so this everybody knows it, this particular thing, dark field microscopy. And in the dark field microscopy, the question comes, which particular light you use? Answer is reflected light. Okay, and in the normal microscope, what we use? Transmitted light. Okay, so this you need to know. The dark field microscopy is using up the reflected light, and it is used for the Treponema pallidum. Fine, and they are spirochete, that's why they look like that, spiral in shape. Fine. And the question recently in the INI-CT was syphilis incubation period, that is 9 to 90 days. What do you get in the primary syphilis? Painless hard chancre. What do you get in secondary syphilis? Rashes over palms and soles. Along with that, condylomata lata. Then comes the latency. Then it goes into tertiary one. Then it is having gummas on the skin. Besides that, along with that, you can have those neurosyphilis and cardiovascular syphilis features. Neurosyphilis stapes dorsalis. Okay. Cardiovascular aneurysm of aorta, ascending aneurysm. What is drug of choice? Penicillin, drug of choice. Okay. And what is the best test as such, most sensitive if I ask you? The answer is FTABS. Most specific, TPI. For the prognosis of syphilis, that is VDRL. VDRL, Venereal Disease Research Lab. This particular test, Venereal Disease Research Lab, this is which type of a test? It is a slide flocculation test. What is flocculation? Flocculation is a type of precipitation. Okay. Precipitation, what is the difference? The precipitation, you use a soluble antigen with antibody. Okay, so this particular soluble antigen is used in that, and there is a precipitate formed. So instead of precipitate, in the case of VDRL, what is produced? Floccules. Okay. Very right. 9 to 90 days. Very right. Wonderful. Now, tell me about this. Which bacterial colonies they look like this particular one? So this is the fried egg appearance of which particular bacteria? This is fried egg appearance of which particular bacteria, guys? Can you tell me? Fried egg appearance of which particular bacteria? Yeah, tell. Fried egg appearance of which particular bacteria? Right, Manisha, you're right. Crystalline penicillin, also penicillin only, na? Which bacterial colonies appears on the solid media? These particular tab. The answer is C, Mycoplasma pneumoniae. Dr. Naveen, you have taken the lead. You're right. Dolly also have told up wonderfully Mycoplasma. The answer is Mycoplasma, and Mycoplasma produces fried egg colonies. Now the winner will be those who will tell up that which other guy does produce fried egg colonies same like this. If anybody tells that, that fellow is scoring here itself about 200. I'm telling off. Which other organism produces fried egg colonies? Let me give you a hint. That's a fungus. That's a fungus. Tell me. That's the fungus. What is the answer? Which produces fried egg colonies like Mycoplasma pneumoniae? Fast. Tell me. ओलिगोडेंड्रोग्लियोमा पूजा ये क्या है? वो क्यों बनाएगा फ्राइड एग कॉलोनीज? शिवांगी तिवारी, you are perfectly right. The answer is Malassezia furfur. Okay, which leads to Tinea versicolor or Pityriasis versicolor. It produces fried egg colonies on the SDA. Okay, on the SDA. And if you are talking about Malassezia furfur only, then you should know spaghetti and meatball appearance, which was the last FMGE and NEET PG question. Spaghetti and meatball appearance is observed when you put those scales into 10% KOH and see them in the microscope. You could able to see those particular short hyphae. Okay, and that is what is termed as spaghetti and meatball appearance. मोदी जी आप बताएं. चिकन वायर की तो बात ही नहीं हुई भाई, फ्राइडे कॉलोनीज की बात हो रही है. चलें आगे. चलो इनके बारे में बात कर लेते हैं. भाई कॉक्सिला क्या करता है? कॉक्सिला बर्नेटी लीड्स टू क्यू फीवर, क्वेरी फीवर, फाइन. एंड दिस पर्टिकुलर वन कम्स इन द रिकेट्सियल फैमिली, दैट यू नो. And Coxiella burnetii is an organism which leads to culture negative endocarditis. Fine. And another important thing regarding Coxiella is that it does not produce any rash. It produces pneumonia-like features. It is the atypical type of pneumonia, and this particular one associates with the cattle birth process and all that. So that you should look out for the history of the question. ये क्या लिखा भाई? आपका आप है किस सर? अच्छा, इंडिविजुअल ऐप आपका. इंडिविजुअल तो नहीं है भाई. दैट इज ऑन द एलन नेक्स्ट नाउ. यू कैन हैव फ्रॉम देयर. द केयूर वधार. राधे राधे, राधे राधे जी. Bordetella pertussis leads to pertussis, also termed as whooping cough, okay, and also termed as barking dog cough. Incubation period here is also 7 to 14. Typhoid is also having the same 7 to 14. Catarrhal, paroxysmal, convulsant, these are the three phases. Borded ganju media is there, bisected pearl appearance, and the drug of choice was asked in the FMG exam. Answer is azithromycin in the case of children, okay. So these are the important ones which you should know. This is also important. Legionella. Legionella guys, Legionella, can anybody tell me what is the culture media for this? The culture media for this is BCYE. What is the full form of BCYE? Buffered charcoal yeast extract. Okay, buffered charcoal yeast extract. Moderate ganju media, it is having bisected pearl appearance. BCYE is the culture media for Legionella, and again Arvind is telling us what is drug of choice. Legionella also azithromycin. Okay, so कहीं कुछ समझ में ना आए ना तो what should you put the answer? Azithromycin. Two drugs are very famous. One is Azi and one is Doxy. Out of them you can do akkad and you can put up. For this also the Azi is the answer. Yeah, very right. Pushpendra, pneumonia, diarrhea. This particular triad is termed as Legionnaire's disease, and this is the pneumonia in which what you have electrolyte imbalances like hyponatremia and all. So that also you need to know. Those who are actively participating now, you all are passing man this time. And let me tell you, those who does microbiology na properly, they are the persons who pass up with a wonderful marks, because they don't know that as such only it's told up that 13-14 questions they come, and if you see of the proper one na, it links to 30 to 40 questions, and 30-40 questions does matter, man. Okay.